This video is adapted from a presentation I gave at iPAK EDU, September 9th, 2024. I'm Josh Middledorf, and my contact information is on this slide. In this presentation, I'll offer my answer to the question, where does aging come from? Why do we age? In particular, the evolutionary perspective that we adopt can and should, I claim, change the way we do medical research on the diseases of old age. Medical gerontology research has been lost in the weeds for 50 years now, and I'll report at the end what I think is a recent breakthrough that offers a near-term possibility of true rejuvenation. There are tantalizing results from rats that need a couple years intensive research before we're ready for human trials. Before I offer you scientific content, I owe you a clarification. How does what I'm saying relate to the journal literature in the field and conventional wisdom? All of the experimental results that I'll offer are well documented in the open literature. However, a lot of the context that I present and interpretation of those results is my own. I've personally been a central figure bringing these ideas into, first, the field of evolutionary biology, and second, the field of gerontology and anti-aging medicine. When I started to write these things 28 years ago, they were radical, but now there are some of the most prominent people in the field who would agree. Nevertheless, my perspective is still a minority for reasons I'll get into. Let's acknowledge that we're not starting from a blank slate. Maybe you've thought a lot about aging and where it comes from, or maybe you've just absorbed some ideas by osmosis. During this presentation, I'll present some ideas that may challenge the way you presently think about aging. Whether these ideas are a better fit with reality than your present way of thinking, that's a decision you'll have to make for yourself. In any case, it's useful to make our assumptions explicit so we can think about them consciously. Perhaps these are part of your thinking, that the body is doing its best to live as long as possible, that natural is better, it's what we were evolved for, that the body is trying its best to be healthy. Let's support the body's natural resilience, that optimizing present health is the same thing as promoting longevity. Here's a question I hope will be thought-provoking. Animals in lab experiments live longer the less they are fed. Those who live longest are on the brink of starvation. Why do you think animals live longer when they're starved? I got my start in this field in 1996 when I asked the same question in a different way. Why should animals die sooner when they get enough to eat? I don't mean obese, just adequate food so that their growth isn't stunted. From this one fact, I formed a hypothesis that the body doesn't want to live as long as possible. If it did, then it would certainly have an easy time doing this with adequate food compared to when energy is barely adequate to sustain life. For the remainder of this time, I want to parse this lesson in three parts. First, what is the evidence that bodies self-destruct with age, that aging is on purpose? I'll talk about why this conflicts with the selfish gene version of evolution, and why so much of the biology community, people who believe so firmly that selfish gene is how evolution works, can't see the evidence for programmed aging for what it is. I'll end this section by responding to the obvious question, how can suicide evolve as an adaptation? How can you reconcile this with survival of the fittest? Next, what are the medical and health implications of this radical idea? What everybody wants to know, how can I improve my chances for living a long time? In this area, my ideas are not so different from what you read from fitness gurus or functional medicine doctors, but I do have my own slant on it. I have an aging website called agingadvice.org. Finally, I'm going out on a limb to tell you about this latest in anti-aging medicine, a technology that I consider to be an extraordinary breakthrough that will not just slow aging, but make us younger. Let's start with the evidence that aging is designed into the animal life plan. In the late 1980s, Tom Johnson was a young genetics researcher at UC Irvine. He worked with C. elegans worms, a favorite nematode roundworm that's convenient to grow by the millions in petri dishes. Johnson grew worms with a defective gene, which he named Age1, after he discovered that worms without it lived 50% longer than normal worms. No one had ever imagined that a single gene could have such an effect on lifespan. In fact, the best experts in evolution had theorized that everything ought to wear out all at once so that no single gene could have any noticeable effect. Johnson's discovery was the more remarkable because longer life required nothing new but deletion of an existing gene. This implied that the effect of the Age1 gene was to cut the worm's life short. What was it doing in the genome? How did it get there, and why did natural selection put up with it? Johnson had a ready explanation. He believed in the conventional explanation for evolution of aging, that is, that aging results from evolutionary trade-offs. Natural selection has had to make compromises, and sometimes genes that cut life short are selected because they have other beneficial effects, especially if they enhance fertility, which is thought to be the very definition of fitness. The worms without Age1 laid only a quarter as many eggs as other worms. It was easy to see how they had been losers in Darwin's struggle. In fact, Johnson's finding looked like a dramatic confirmation of the theory that aging was a side effect of genes for greater fertility, greater individual fitness. Aging had not evolved directly, selected for its own sake, but as a side effect of greater fertility, so paradox avoided. But a few years later, this story turned on its head, and what had been confirmation of evolutionary theory became a direct contradiction. Johnson discovered that his mutant worms actually had two genes that were different. In addition to Age1, there was another unrelated gene defect, FUR15, on a separate chromosome. By crossbreeding, he was able to separate the two. Worms with the FUR15 mutation had impaired fertility, but no extended lifespan. Worms with the Age1 mutation had extended lifespan with no impaired fertility. This was a full-fledged Darwinian paradox. The Age1 allele that was found in nature was the one that gave the worms a short lifespan. It was the defective gene that caused the worms to live longer. Age1 looked like not a selfish gene, but an aging gene, a suicide gene. It was just the kind of gene that natural selection ought to eliminate handily. How had this gene survived, and what was it doing in the worm genome? Johnson published this second paper on Age1 in 1993. Since then, there are many, many genes that have been discovered that shorten lifespan, not just in worms, but in fruit flies and even in rodents. Remove the gene, the animal lives longer. For some of these genes, there are auxiliary benefits. The word is pleiotropy. But for about half of the genes, no pleiotropic benefit has been discovered. They seem to be aging genes, pure and simple. This is the most direct evidence we have that aging is an evolutionary adaptation. Nature wants us to die on schedule. It gets better. Some of these genes are common to all eukaryotes, that is, all multicelled life forms and even some protozoa. That means that aging genes have been conserved by evolution for at least 1 billion years. Any gene that's common to all animals has been conserved by evolution, and we can safely assume it must have a core purpose. Genes for copying and transcribing DNA, genes for the Krebs cycle are highly conserved. They're centrally important to life, so evolution doesn't want to monkey with them. They're in a protected portion of the genome with a very low mutation rate. Evolution has placed aging and death in this same category, conserved mechanisms essential to life. The best example is insulin. You probably think of insulin as a hormone that regulates blood sugar. You're right about that. But worms have insulin, and they don't have blood, let alone blood sugar. Yeast cells have insulin. We've learned that insulin has a much deeper role, an older role. Insulin is a regulator of lifespan. In yeast cells and in worms, insulin is expressed when there's plenty of food and it shortens the lifespan of the yeast or the worm. Regulating blood sugar is an afterthought that evolved much later. I told you the story of age 1, the first aging gene that was discovered in 1990. The second was discovered by Cynthia Kenyon working at UCSF in 1993. That gene was called DAF2. When she mutated the DAF2 gene so it is barely functional, the worm lived twice as long. So of course her next move before she even published the paper, she had to find out what is DAF2 all about. She found that it's an insulin receptor. DAF2 is the only insulin receptor in lab worms. This was our first hint that generally in most animals, insulin regulates lifespan. In fact, we know that in humans, type 2 diabetes is intimately connected to aging. As we get older, we lose insulin sensitivity. If we're couch potatoes, we lose insulin sensitivity much faster. The pancreas pumps out more insulin, and then we're more likely to get Alzheimer's disease. We're more likely to get cancer. Our life expectancy is shorter the more insulin is in our bloodstream. There are other examples in nature of what looks like deliberate self-destruction. In mammals, like ourselves, there are four principal mechanisms by which the body destroys itself as we get older. They are inflammation, telomere attrition, autoimmunity, and apoptosis, which is self-suicide. All of these mechanisms are essential for life, but as we get older, the mechanisms are repurposed for destructive ends. They start out protecting us, but end up by destroying us. It's fair to ask, does that mean that aging is an inevitable consequence of having these functions that are good for us early in life? Or maybe they just become defocused or lose their specificity over time? I don't think so. When I look at these, it's obvious to me that the self-destruction is a purposeful adaptation. They're not mistakes or dysregulation. Each of these is a protection function that's repurposed later in life. And this change in function looks to be programmed and not inevitable. But I'll leave it to you to decide. We'll take the four in turn. Let's take them in turn. The first is inflammation. Inflammation is the body's first line of defense against invaders. Strong chemical oxidants are produced that degrade tissue and kill pathogens. When you cut yourself, the cut becomes inflamed, both to destroy the damaged tissue so it can be replaced and to protect against bacteria that might enter the cut. But when we get older, inflammation becomes systemic. The regulation of inflammation changes so that inflammation damages the body everywhere. Inflammation in the brain is a primary cause of dementia. Inflammation in the arteries is a primary cause of heart attacks and stroke. And inflammation systemically is associated with carcinogenesis. Anti-inflammatories such as aspirin and turmeric and fish oil are some of the most reliable life extension supplements that we've got. Second is telomere attrition. You've probably heard of telomeres. They were popular as a theory of aging maybe 15 years ago, and they were subject of the 2009 Nobel Prize. Briefly, every chromosome has a tail of repetitive DNA that carries no information but protects the ends of the chromosome from damage. Every time the cell divides, the chromosome is replicated, but the enzyme that copies the DNA fails at the very end. The telomere gets a little shorter with each cell division. Eventually, the telomeres get critically short, and the cell goes into panic mode called cellular senescence. These senescent cells are few in number, but they do great damage. They broadcast signals that increase inflammation. For example, there's a group at the Mayo Clinic that conducted research about 15 years ago in which they genetically modified mice so that there was an artificial marker on all the senescent cells. These mice were engineered so that a drug could be given to them that would kill only the senescent cells and leave all the normal cells intact. When senescent cells were eliminated, there was a dramatic increase in lifespan, up to 30%, even though the mice were treated late in life. This proves that senescent cells are an active mechanism of aging. In practical terms, their research launched a search for agents that could selectively kill senescent cells in humans who have not been genetically engineered. Such drugs are called senolytics. The challenge is that the number of senescent cells is small, the number of normal cells is much larger. In order for a senolytic drug to be effective, it has to have a very high selectivity, so it's only killing senescent cells and not normal cells at all. We're working on it. The best thing we have so far is a combination of supplement called quercetin with a chemotherapy agent called dasatinib. But fasting for more than 3 days is senolytic. Perhaps the main reason that fasting is healthy is that it kills senescent cells. Most importantly, senescent cells in the immune system. Walter Longo of USC has built a whole system of cancer treatment and life extension on this finding. I like to take quercetin or fisetin when I fast. I stay away from the chemo agent though. Third is autoimmunity. Our immune systems are brilliantly able to distinguish self from invader. We have T cells that label invading bacteria or foreign cells and mark them for self-destruction. Obviously, we don't want the body's immune system turning against our own cells, and yet we have tens of thousands of different proteins, hundreds of different types of cells. The immune system is a miracle of nature for its ability to reliably distinguish self from invader. It's so good that if we get a kidney transplant from a family member whose DNA is 99.9 plus percent identical to our own, the immune system detects the difference and goes after it. Not only that, but a cancerous cell or a cell infected with a virus may produce proteins that the immune system recognizes as not belonging to self. Then the immune system goes to work destroying our own cell before it can do more damage. The immune system is our primary defense against cancer as well. We're destroying potentially cancerous cells every day of our lives. One of the problems with the mRNA shots is the concept of producing the antigen in our own cells. The body detects that the cell is producing a foreign protein and eliminates not just the protein, but the entire cell that has taken in the RNA lipid nanoparticle. Then the problem is that sometimes the body learns the wrong lesson in the process and starts attacking other similar cells, even though they're not infected by the lipid nanoparticle. So mRNA vaccines can cause autoimmunity. Autoimmune diseases are lupus and variants, including arthritis and multiple sclerosis. They increase in prevalence late in life. Part of the explanation for autoimmunity is the shrinking of the thymus gland. The thymus gland is a little organ the size of your thumb that lives at the top of the rib cage. The T cells are so named because they are trained in the thymus. The thymus trains T cells to distinguish self from invader. It's most active in children, and the thymus gland begins to shrink before we're 10 years old. By the time we reach age 70, there's very little left of the thymus, and what we have is mostly non-functional fat cells. Some anti-aging strategies target the thymus for regrowth. There's a protein called FOXN1 that stimulates thymus growth, but it's hard to get and it's easily destroyed in the body. My friend Greg Fay has a program for regrowing the thymus using a cocktail of 5 supplements, one of which is growth hormone. But growth hormone has its own problems, and on its own, growth hormone can shorten lifespan. If you want to know more about the TRIM study, Google TRIIM. Fourth is apoptosis. Apoptosis is programmed cell death. Cells have the ability to self-destruct when they detect that they've become a threat to the organism, or even when they're not needed. Cancer cells self-detect that they are cancerous, and they fall on their swords. The trigger for this is a protein called P53. The executioner is the mitochondria. Those organelles that create energy for the cell are expert in the high energy chemistry of reactive oxygen species. Apoptosis is important in development too. The hand develops when vestigial cells between the fingers commit suicide. Even our brains developed with too many neurons, and the excess neurons prune themselves, somehow leaving a functioning brain. But as we get older, apoptosis is on a hair trigger. The balance of signals that promote apoptosis and inhibit apoptosis is shifted in favor of the former. So we lose healthy functioning muscle cells to apoptosis. This is called sarcopenia. And we lose nerve cells in the brain, which can be a contributing factor to dementia. I mentioned Walter Longo a little while ago. In the 1990s, Walter was a grad student doing his dissertation on apoptosis in yeast cells. He found that when a colony of yeast cells was starved, 95% of them would undergo apoptosis, turning themselves into food for the remaining 5%. This is a good trick, a survival strategy for the colony. But the individual cells that decide to kill themselves are making the ultimate sacrifice. Evolutionary theory predicts that everyone would want to be part of the 5%, and none of them would volunteer to be in the 95. Based on theory, Walter's thesis was rejected for publication time and time again. They kept demanding more lab results to prove that what he was seeing was really apoptosis. By the time it was published in Nature in 2004, the case was rock solid, and it stands today as one of the best examples that we have that programmed death can be an altruistic adaptation. I want to continue now with more reasons to believe that aging is programmed under the body's control. This one is under the category Fun and Amazing Facts About Nature. There's no rhyme or reason to lifespans of animals. Lifespans do follow patterns, of course. Larger animals tend to live longer. Animals protected from predators by flight or burrowing or climbing trees live longer than animals that crawl on the ground. But if you look within categories, it becomes clear that lifespans are adjusted to ecologies. There is no physical necessity, no limitation, or even necessary trade-off. Here are some examples. Mice typically live for 2 years and they die of cancer. Bats are the same size, but they burn much more energy. They generate a lot more free radicals. You might think they'd burn out, but no, bats typically live for 40 years or more. Rats live for 2 or 3 years typically. Squirrels are the same size, similar activity level, but they live for 20 years. Yes, it's true that rats are much more likely to be caught by foxes because they can't climb trees. But if they're not caught by a predator, they still die of old age before age 3. Crabs can grow to a fixed size and then they die in 2 or 3 years. Lobsters just keep growing and growing and growing, probably without limit. You can say that they age backwards in the sense that the older they are, the less likely they are to die. The older they are, the less likely. That's the opposite of you and me. Google found for me this picture of a lobster that's 132 years old. Now we come to my first clue that got me interested in aging in 1996 and convinced me that animals are not evolved to live as long as possible. I came to learn that for many kinds of animals, not just lab mice, they live longer when deprived of food. Later I learned to think of this as hormesis. I don't know if you know the word hormesis, but the concept is probably familiar to you. We're healthier if we're challenged. Nietzsche famously said, "What doesn't kill me makes me stronger." You probably know that exercise is good for your health, but have you ever thought about why? Exercise generates free radicals. Exercise makes micro tears in muscles. Exercise uses energy that the body otherwise might use to repair itself for autophagy and error correcting DNA. Why should exercise make you healthier? Starvation, of course, is an even more extreme example. In the lab, the less an animal is fed, the longer it lives. What is the body able to do when it's starved to the brink of death that it couldn't do if it had enough to eat? You can extend the lifespan of rats by chilling them in cold water, or you can overheat them. They also live longer. Heat shock protein is the name of an enzyme that is generally protective. There's a series of studies out of Finland showing that people who take regular saunas have reduced cardiovascular risk. Radiation, hormesis, is also a thing. Low-level radiation extends life of lab animals. Some of the survivors of Hiroshima lived unusually long. Certain toxins have a similar effect. Low doses expend lifespan. You can extend the lives of lab worms by exposing them to Agent Orange, which is a powerful pro-oxidant. In this case, the quantities are not necessarily low. The worms suffer damage and yet they live longer. Contrast hormesis with homeostasis. All living things are homeostatic, which means if you put them in an environment with any challenge, they adapt to that challenge and suffer less damage than you might think if they were completely undefended. But the paradox of hormesis is that living things can overcompensate. They adapt so much that they actually do better in the damaging environment than in a benign environment. So why don't they do this all the time? Why should an animal live longer when it's starved or poisoned or when it's overheated? Maybe we should be asking the converse question. Why do animals have shortened lifespans when they have everything they need and life is easy? The clear conclusion, the conclusion that evolutionary biologists don't want to face, is that animals are evolved to live for a programmed amount of time. The program is flexible. If the environment is challenging, say there's a famine and lots of others are dying of starvation, then programmed death backs off. But if conditions are favorable, there may actually be a risk of overpopulation, and then programmed death goes into high gear. This is our first hint that aging is a form of population regulation. Why should this be controversial? I'll mention parenthetically here one of Britain's most eminent naturalists, a profoundly learned man who keenly observed and described nature in eloquent prose. This man was V.C. Wynne Edwards. In the 1950s and 60s, he was writing about population regulation in Nature. We know from experience during COVID how a scientist can be canceled if he opposes the politically correct narrative. Well, 50 years ago, this is exactly what happened to Wynne Edwards. The hit job was done by evolutionary theorists, people who knew some mathematics and understood selfish gene theory well. It wasn't called selfish gene theory then, it was called population genetics, and it was evolutionary orthodoxy. It was the only evolutionary theory that most students learned in grad school. So they came after Wynne Edwards with a vengeance. They ridiculed him for not understanding that it doesn't matter what he saw in nature. What matters is the mathematical theory of population genetics. And the mathematical theory says that population regulation is not a thing. You fool! Survival of the fittest. What part of survival don't you get? Evolution is all about maximizing reproduction, not regulating populations. You can't regulate populations without compromising reproduction. That's the opposite of fitness. The buzzword in this controversy was group selection. Does natural selection choose what is good for the individual or what's good for the community? Theorists claim that group selection ought to be much weaker than individual selection. Theory says that natural selection works at the level of the individual, so that if it's good for the group but bad for the individual, it can't evolve. Kin selection is real. We can understand that if a mother sacrifices her own life to save two or more offspring because they share her genes, this represents an evolutionary advantage. Her genes will prosper. But if an individual sacrifices for the sake of a larger community that's only distantly related, that's not a thing. It can't happen. It would require group selection. In science, observation should always take precedence over theory. So I'm sorry to say that in this case, theorists won the argument. Wynne Edwards's reputation was trashed. A younger generation of mathematical theorists took over the field. The best of them were Bill Hamilton, John Maynard Smith, and George Williams. In recent times, there's been some backlash against these guys, but their way of thinking and the selfish gene idea are still dominant in the field. I come up against them every time I try to publish a paper. Some of the more successful people in the field have confided to me that they soften their language. They avoid talking about group selection because they know that it will hold up their paper in peer review. So I'm one of the few scientists who is constantly in their face, raising examples where you need group selection to explain what we see in nature. The selfish gene is just one small part of the way evolution works. There are others much more focused and much more influential than I who have also done battle against the selfish gene ideology. I'll mention George Price, Lynn Margulis, David Sloan Wilson, Michael Wade, Charles Goodnight, Michael Gilpin, and John Pepper. I mentioned Bill Hamilton earlier as one of the selfish gene theorists. Early in his career, he laid the foundation for the theory of kin selection, and he's known for Hamilton's rule, which is the formula for balancing the cost to an individual doing a certain action against the benefit to kin from that same action. But Hamilton was a naturalist, and so he saw examples in nature that couldn't be explained by Hamilton's rule. Later in his life, he expanded his view of how evolution works in a book called Narrow Roads of Geneland. He died tragically on a safari in Africa, so he missed the chance to participate in the deconstruction of the selfish gene. George Price, in the upper left, extended kin selection to create a mathematical theory of group selection. His life is an idealistic and tragic story. Look him up if you're interested. David Sloan Wilson was my mentor when I came into the field. He graciously co-authored my first paper and introduced me to the academic community. His entire career, beginning in the 1970s, was devoted to what he called multi-level selection, MLS. Evolution operates at the level of the gene and the individual and the group and the community, and different traits are best explained at different levels of selection. He's comfortable both with experiments and mathematical theory. Wilson is a talented writer both for academic journals and in a series of popular books. Michael Gilpin introduced computer modeling into the field in 1975. Part of the reason that the selfish gene illusion was allowed to take hold is that the math of selfish genes could be simply expressed in equations that you solve by hand. The theory was developed in the 1920s, before computers. Group selection is evolutionary ecology, and for this you need computer models. In 1975, Gilpin published a monograph called Group Selection in Predator-Prey Communities. This book built a theoretical foundation under Wynne Edwards's ideas about population regulation. It's really quite a lesson in the way that computer models should be used. He explains the assumptions of each step at the model, and he argues that the assumptions that he makes is conservative in the sense that reality must be more friendly to group selection than the assumption that he's making. Nevertheless, Gilpin's book was not well received. He stepped on too many toes. Also, computer modeling was too new, and the theorists didn't trust it. Gilpin had a short academic career before he moved to Montana, where he worked in wildlife conservation, theory and practice. Gilpin's insights are the basis for my theory of aging, which is about demographic homeostasis, a fancy word for population regulation. Evolutionary theorists have moved on, and Gilpin is all but forgotten. So I sing his praise every chance that I get. When I published my book, I tried hard to find him, to give him a copy. I have two friends who knew him back in the old days. If you know where Michael Gilpin is, please put me in touch. Even these people, Wilson and Gilpin and Wade, don't talk about aging as a group-selected adaptation. Aging is particularly hard to explain because the cost to the individual is death itself. That is, complete loss of future ability to reproduce. And the benefit is widely distributed, not just to kin, not even confined to the same species. The benefit is to any others that share the same prey. And here's another footnote. Some people say there's menopause, there's andropause. By the time an animal has died of old age, the ability to reproduce has already diminished or disappeared. So evolution has nothing to lose by letting that individual die. Yes, that's true as far as it goes. But why do animals lose their fertility as they age? That's the question. It's not a biological necessity. Trees grow larger and produce more seeds, and they continue to do this for decades, sometimes for centuries. They get more fertile with each passing year. There are a few animals that can do this too. Clams get bigger and bigger. 6 foot, 500-year-old clams can lay millions of eggs. Lobsters and sharks also grow larger and larger without limit, and they lay more eggs with each passing year. So the question is, why has natural selection created reproductive senescence? Or why has evolution allowed this to happen at such great cost to the individual and marginal benefit to the community? Hamilton's rule, as extended by George Price, can't answer this question. Wilson's multi-level selection also can't explain aging. Only Gilpin's model works to evolve aging. My contribution to this field, my theory of aging, is built on population regulation as described by Wynne Edwards and as first modeled by Gilpin. Since I'm diverging from the evolutionary orthodoxy, I owe you an explanation. How is it that the evolutionary orthodoxy became the evolutionary orthodoxy? And why don't I believe it? So here's a bit of the history. Charles Darwin was a naturalist. His theory of evolution wouldn't qualify as a theory in the modern sense because it's descriptive, loosely worded, and it makes no predictions. In 1900, the work of Gregor Mendel, a contemporary of Darwin, was widely available to the scientific community for the first time. Mendel had decoded the rules for inheritance. There was an opportunity to combine Mendel with Darwin to make a fully quantitative predictive theory in the spirit of Newtonian physics. The person who picked up this challenge was R.A. Fisher, Ronald Fisher. Fisher was a brilliant mathematician who's responsible for much of the methodology that we take for granted in statistics today. He was also a flaming eugenicist, alarmed that the poor were having too many children and the rich not enough. Fisher's mathematical theory of evolution is variously called population genetics or neo-Darwinism or selfish gene theory or just the new synthesis. The attraction of his formalism is that it's very explicit, derived from postulates like Euclid. 3 generations of evolutionary scientists have built on his theory. In the Fisher formalism, there are non-overlapping generations, the environment is fixed, population size is constant, presumably because competition enforces a fixed carrying capacity. Every gene is correlated with a trait, and the trait is associated with a linear contribution to fitness. If the trait leads to more copies of the gene in each successive generation, then the trait spreads through the population and eventually takes over. If the trait leads to less copies, it rapidly dies out. That's the theory in a nutshell. Fisher's neo-Darwinist formalism makes possible a wide range of predictions. The downside is that none of these predictions hold true in the real world. How then has the theory thrived for 100 years? The answer is that the predictions have held up consistently in lab experiments. Evolutionary scientists do breeding experiments in the lab, and the results are fully in accord with Fisher's predictions. This is taken as validation that the theory is correct. But there are glaring contradictions between neo-Darwinist predictions and what we see in the real world. Of course, I'm talking about aging, but there's much more. The evolution of sex, many other cooperative behaviors, ecological commensalism, and the development of complex traits that could never have been built one mutation at a time. Vision with complex eyes is commonly cited as an example. Here's the connection to aging. The mainstream of evolutionary scientists are neo-Darwinists. According to their paradigm, aging makes a negative contribution to fitness, and so it should die out. It should never evolve. Over the years, a hodgepodge of incoherent ideas has grown up to explain why, nevertheless, almost every animal in nature suffers aging and obligate death. The first such idea, attributed to Peter Medawar, was that aging just doesn't matter because in the wild, no animal lives long enough to die of old age. But when actual field studies were done, 30 years after Medawar, this turned out not to be true. The second, attributed to George Williams, was that the genes that cause aging are really fertility genes. Aging is an inseparable side effect of genes that increase reproductive fitness. This was proposed before there was a science of epigenetics, in which we know that genes are constantly being turned on where needed and turned off when not. A third theory from Thomas Kirkwood was that the body devotes so much food energy to reproduction that there isn't enough left over to do an adequate job of keeping the body in good repair. Well, this obviously contradicts the fact that animals live longest when they're starved. Then there's the wear and tear theory, vaguely invoking increasing entropy, loss of information, the second law of thermodynamics. But living things take in free energy from the outside, and so there's no need for them to suffer increasing entropy as they age. If the second law applied to living things, they would never be able to grow from a tiny egg to complex and fully functional adults. My role in this field has been to face squarely the fact that evolution has arranged for us to die on a schedule, and that none of the explanations within prevailing theory can account for this fact. So, what's my theory of how evolution can prefer programmed death to unlimited life? Every living thing depends on an ecosystem to live. No one lives in isolation. This was seldom appreciated by early mechanistic biologists, but it is absolutely true. Turns out you can't build an ecosystem if everyone's trying to live as long as possible and have as many babies as possible. We need both birth control and death control from all constituent species above the lowest trophic level if we are to build a stable ecosystem. The problem, as cited by the neo-Darwinists, is that individual selection is direct and rapid, whereas if a trait only benefits the group at the expense of the individual, it first has to evolve uphill against individual selection. It has to take over the group before natural selection can prefer one group to another. This is a slow and improbable process. But all this is based on Fisher's standard assumption that the group doesn't change in size from one generation to the next. As Gilpin demonstrated 50 years ago, a population can expand very rapidly, and within just a few generations, it can expand way beyond its sustainable capacity and crash in extinction. This is a rapid process that can easily compete with the neo-Darwinist mechanism of changing gene frequency. So Understanding ecological relationships and population dynamics leads to a hypothesis for how aging evolved as an adaptation on its own, not linked to fertility or anything else. This is my academic book on the subject, covering evidence, theory, and implications for medicine, fully footnoted. The in-your-face title is Aging is a Group-Selected Adaptation. The cover cartoon is from the Tortoise and the Hare fable, with Darwin as an old man as the tortoise, and Darwin as a young man, the hare. And this is a popular book on the same ideas, supplemented with the storytelling of Dorian Sagan, an interesting character and well worth getting to know. Now I want to take this theory as a foundation and talk about why it matters. The way we stay young, the way we research medical interventions for life extension depend on what we think aging is and where it comes from. When I started in this field in the mid-1990s, the study of aging was a backwater, with a few curious souls studying the causes, but the only known life extension strategy at the time was starvation. There was no field of anti-aging medicine. It was widely perceived, based on mythology more than science, that aging was inevitable. Some people thought aging was inevitable because it's a physical process akin to the accumulation of entropy. This theory is a non-starter, as I said. Others believe that evolution has been working for a billion years to make lifespan as long as possible, so there's little that humans can realistically expect to do to improve on that. But we should not be so fatalistic once we realize that evolution has affirmatively arranged for us to age. Maybe all we need to do is to throw a monkey wrench into the gears of the self-destructive mechanisms that nature has endowed us with. There are examples in nature of living things that don't age. There are many plants that don't age, and a few animals. I mentioned clams and lobsters. There are jellyfish and dung beetles that are capable of devolving from old to young and starting life over again. There's nothing inevitable about aging. It is properly the subject of evolution, not of physics. Well, all that has changed now. There's venture capital in the field, trying to make money on anti-aging pills. But most of the projects are doomed to fail because they have the wrong idea about aging still. They're not thinking clearly if they're thinking at all. They look at aging as a cellular process. Cells lose their integrity or they're becoming dysregulated, or their waste products accumulate, or their DNA has copying errors, or their gene expression wanders off in some random direction. What they fail to realize is that aging is not a cell-level process, but a systemic process. It is organized from the top down, not imposed from the bottom up. The only successful anti-aging strategies so far work with the body's signaling chemistry. Trying to repair cells from the bottom up is a fool's errand, but modifying the signals in the blood that instruct the cells to be young or old, this is a promising avenue where some dramatic success has already been achieved. A lot of the alternative doctors out there, whom I like and agree with about most issues, fail to realize that anti-aging is essentially different from optimizing health. To optimize health, we work with the body, natural foods, clean air, avoiding toxins, good nutrition, including micronutrients. This approach will help you to live out your natural lifespan, but it won't extend your natural lifespan. To extend human lifespan, we have to play tricks on the body. The things we do are the opposite of natural. The first level of tricks we can play involve the built-in flexibility of the aging program. We're evolved to live longer in harsh environments and shorter in cushy environments. So we trick the body into thinking that life is tough. This is hormesis, the concept I mentioned earlier. The best hormesis strategy is fasting, but there are others, including exercise, exposure to heat in the form of saunas, cold, maybe ice baths or cold showers, hypoxia, breathing low oxygen air, maybe low dose ionizing radiation, maybe even some toxins, alcohol in very small quantities. Jeanne Calment is the only person documented to live past 122, and she reportedly smoked 1 cigarette per day. There are some supplements that your doctor may not tell you about, but that are worth adding. Melatonin not only helps you to sleep at night, which in itself is important for health and aging, it's also an anti-aging hormone working through the mitochondria. Likewise, high-dose vitamin D can add a couple of years to life expectancy. Anti-inflammatories help. Curcumin, omega-3 fats, aspirin, boswellia. Rapamycin is a drug that works consistently in mice, adding 20 or 30% to lifespan. In humans, the timing and dosage have not been worked out, but there are lots of people experimenting. Metformin helps maintain insulin sensitivity. Berberine and gynostema are herbal alternatives to metformin. The only downside is that they may compete with exercise for the same benefits. DHEA is another hormone that helps with insulin sensitivity and extends lifespan in mice. NAC, N-acetylcysteine, is a personal favorite of mine. It's a precursor of glutathione, which is an essential peptide for mitochondria. It extends life in mice almost as much as rapamycin, and it has no downside. Selegiline is a prescription antidepressant that has been known to extend lifespan in mice, though data is thin. Spermidine is a small molecule that offers dramatic life extension in rodents, but at doses that are much larger than you can get in commercial pills. There's a peptide called epitalon, which has shown great promise in Russian experiments. The strategy that's missed in a lot of anti-aging books involves psychosocial factors. We're all social animals, and evolution has provided that important people in the community get a longevity boost. Live an active, fulfilling life. Be empowered and creative in your community. Maintain close, loving ties with friends and family. Engage with politics. Be physically close and sexually active. These factors are worth about 10 years of life, comparable to the difference between a pack-a-day smoker and a non-smoker. Social factors have far more effect on your life expectancy than the supplements you take or whether or not you eat organic food. Doing all of these things well gets you to about 90 or 95 years of life expectancy. Beyond that, your genes take over. You can't live past 100 unless you have centenarian genes. Or if we hack into the body's aging program and trick the central signaling mechanism into thinking that you're not really old. This brings me to the cutting edge of anti-aging research. The idea that the body knows how old it is and gradually destroys itself on schedule implies that there's a clock in the body that measures years. Certainly this is true since we know that growth and development are timed. Sex hormones reliably emerge at puberty. It's probable that the same clock continues to function as we get older, and it controls the timing of inflammatory cytokines and pro-apoptotic signaling, for example. We don't know where that clock is or how it works, but we have a good idea how information about age is transmitted through the body. It is transmitted through the bloodstream via natural lipid nanoparticles called exosomes. Exosomes are virus-sized droplets of biologic information enclosed in a protective layer of lipids, which are just fats. They seem to be a universal biological language that crosses species barriers and carries information about all kinds of things. Exosomes are exchanged by plants in the forest to warn of an invading insect pest. Exosomes are breathed out by people who encounter a new virus or bacteria. The exosomes are then breathed in by others, and their immune systems get a jump start on infections that might be in the neighborhood. This is just one of the reasons why isolating people during COVID was such a bad idea. We now have evidence that exosomes in the bloodstream carry information about the age of the body. Exosomes tell every cell in your body to act like a 20-year-old or act like a 40-year-old or a 60-year-old. We have preliminary data with rats. Three different laboratories in Mumbai, India, in Los Angeles, and in Nanjing, China, have all reported that they are able to make old rats into young rats using exosomes. Two of the laboratories used exosomes from young rats. One actually used exosomes from young pigs to rejuvenate old rats. This is really encouraging because it suggests that exosomes can cross species barriers to transmit information about age. We're not ready for human trials yet, but almost. There's a few years of lab work yet to be done, and we need a scalable way to extract exosomes from young pigs or perhaps from cells cultured in a petri dish. Exosome therapy is far and away the most promising area of anti-aging research today. It's being held back by economic factors. You can't patent exosomes. It's not clear how the people or companies that invest in this research will have IP rights to a product that they can sell later on. For the sake of science and for public health and for the future, exosomes should be a research priority for public funded laboratories. Thanks for listening, and thanks for being open to new ideas.